Good morning. Welcome to the webinar on Clarity-Bpa. My name is Pete Myers. I'm an adjunct professor at the Institute of Green Sciences at Carnegie Mellon University and also chief scientist at Environmental Sciences. This webinar is about Clarity-Bpa. Clarity-Bpa is a visionary collaboration between the U.S. National Institute of Environmental Health Sciences, the US FDA, the US National Toxicology Program, and 14 independent academic laboratories at institutions around the country. Clarity was founded to help resolve a major conundrum facing efforts to determine whether low doses of BPA, those experienced by many people in the US and around the world, are safe. Guideline studies conducted by industry for regulatory authorities report that those levels are safe. But many studies from university labs around the world and some government agencies in Europe find differently. Our first speaker, Dr. Gerald Heindel, worked until his retirement at the US National Institute of Environmental Health Sciences as the extramural program director overseeing research grants from NIEHS on endocrine disruption. He became very concerned that the work of leading BPA experts funded by NIEHS was not being used in any meaningful way by US regulatory agencies to make determinations about what doses were safe and which were not. He realized that a new approach, which he called Clarity, could solve this conundrum. He's going to tell you what Clarity is, why we needed it, and then introduce the rest of the panel, 3 of the world's leading experts on BPA's low-dose effects. Dr. Heindel.
Thank you, Pete. After all this practice, sorry about that. My goal here is to give an overview of the BPA Clarity. And the bottom line is to set the stage for the rest of the talks today. So Clarity-Bpa is a consortium linking academic and regulatory insights into the toxicity of BPA. It's a new collaborative model, and the goals of this model are to integrate the strengths of a guideline study with the molecular and disease-associated endpoints from independent scientists into one study. And hopefully that extra data will help to improve risk assessment. And as Pete noted, this is a very complex study with partners from the NIEHS, the FDA, and 14 NIEHS-funded grantees. So why did we come up with Clarity anyway? What's the problem that we developed Clarity to solve? And that's shown on this slide. When regulatory agencies look at the data they have that they can use to make risk assessments, they come in 2 types: guideline studies and independent scientist studies. The guideline studies have a very standard design, standard endpoints, and the whole study design and endpoints are government approved. There's usually only 1 or 2 guideline studies. On the other hand, there can be many hundreds of independent scientist studies, but they have a very diverse design. They can be cell cultures, zebra fish, mice or rats. They have a wide variety of endpoints, from molecular endpoints to disease-associated endpoints. And in many cases, they have far fewer animals because that's fewer animals are actually needed for their studies. So when it comes to the regulatory agencies looking at this data, they tend to focus more on the data from the guideline studies and not the independent scientist studies. That seems to be a problem because a lot of time and effort and money is put into these independent scientist studies, and it's important that they be useful for regulatory agencies. So we came up with a way to potentially solve that, and that's the Clarity approach. And the idea then was to merge the guideline study and independent scientist study into one coordinated, integrated study where the guideline part of it does all the animals, and then the animals are shared between the core aspect of the guideline study and the independent studies. So you have a core study, which is usual to the guideline study, and independent studies from scientists. Then this overall design is basically a government-approved design, and it would provide a more comprehensive assessment of toxic effects. So that's the Clarity design. So here's the overall approach then. It's a little different than a typical guideline study, but a lot of it is the same, where you have a continuous dosing starting on gestational day 6 all the way through 1 year and 2 year. But in this case, we added a stop dose where you start dosing at the same time, but stop dosing at postnatal day 21 and let the animals grow up to be 1 and 2 years of age. And all of the dosing is done by gavage. So for the FDA core study, they look at results from the continuous dosing at the end of 1 year and 2 years. And the independent scientist studies look at results depending upon the study from either the continuous or the stop dose study, but looking at a variety of different time points, starting at postnatal day 1 up through 1 year. The Sprague-Dawley rat was used for these studies. This is the Sprague-Dawley rat that's used for the guideline studies at FDA. And the FDA core study part had 50 animals per group. The independent scientist studies had 10 to 12 animals per group, whatever number was required to give statistical significance for their studies. There was a vehicle control and 5 doses of BPA from 25,000 micrograms per kilogram per day down to 2.5. And you should note that the 2.5 microgram dose is in the range of human exposures. Now, since at least part of the effects of BPA are thought to be due to the estrogen receptor, we added a positive control, ethinyl estradiol, to show that these animals would indeed be sensitive to estrogens. And that was done at 2 doses. And as I mentioned, the route of administration was gavage, gestational day 6 to birth, and then starting on postnatal day 1, the pups were dosed up until postnatal day 21 and then stopped, or all the way up through 2 years. These are the endpoints. The FDA core study endpoints were the typical endpoints from a guideline study, a variety of clinical chemistry endpoints, some male and female reproductive assessment, histopathology, and organ weights of tissues. On the other hand, we have the endpoints of the independent scientists, and you can look and see A variety of different tissues were examined for molecular and disease-associated endpoints. Now, these 14 grantees were all chosen because they had shown in their publications that they could show BPA had effects on these particular endpoints. And the goal was to look at these endpoints now in this Clarity-Bpa integrated protocol. So the study is now finished and publications are coming out from the independent scientists. There's about 8 or more publications now out. And over the next few months, we expect that all the data from the independent scientists, whether positive or negative, would be published. And you also know that the FDA core part is being published. What we're still waiting for is the final integrated report that covers the results of both the core study and the independent scientist study. And that integrated report should cover not only the data, but a detailed analysis of the data, data interpretation, and lessons learned. So it's important as you hear people talk about results from the BPA Clarity study, that many times they're talking about results just from one part, either the independent scientist or the FDA core part. Since there's no BPA Clarity total report, and that report is not out yet. So you can't call it a Clarity-Bpa report until we have this final report finished. So that's my overview of Clarity, what it is and why we were doing it. And now we're going to move to 3 separate presentations. The first one by Laura Vandenberg, who's an expert on endocrine disrupting chemicals, focusing on low dose and non-monotonic dose response curves. And she's going to talk about some positive results from the FDA core part of the study. Then Gail Prinz, who's an expert on prostate cancer and endocrine disrupting chemicals in prostate cancer, will talk about some significant results from the independent scientist studies. And finally, Pat Hunt, who's a germ cell biology and toxicology expert, will pull it all together and talk about lessons learned. So now I'll turn it over to Laura Vandenberg.
Thanks, Jerry. I'm going to speak to you about my analysis of the data that was published in the core study. Of course, noting that I was not involved in the Clarity study, so this is a view from outside of the study, using the data that is publicly available through the FDA's draft report. And as Jerry noted, and I just want to give you a brief reminder, for each organ that was evaluated, there are guideline outcomes, usually weight of organs and histopathology of organs, and then academic outcomes. And Dr. Gail Prinz will be talking about those academic outcomes. So just a very brief overview or broad overview of the results from the FDA core study. This comes from an analysis that I've done with Pat Hunt and Andrea Gore, and looking at each organ system, looking at whether there were significant effects of BPA at any of the doses, demonstrates that there were significant effects of BPA in both males and in females, and that those effects were seen across the range of doses. I'd like to talk about some of the serious effects of BPA that were observed in the FDA core study, and in particular, effects that were observed at low doses. At low doses, there was an increase in mammary cancer. I'll talk about this in a second. There was an increase in inflammation of 2 lobes of the prostate. I'll also talk about that in a second. There was also kidney nephropathy. This is altered kidney function and health at low doses. And there was obesity in adult females at later ages of life at one of the mid doses. Again, we consider this to be in the range of low doses. So looking specifically at some of the data, this is looking at mammary adenocarcinoma. This is a cancer of the mammary gland. These were animals that were exposed to BPA only during early development. And what we see is that in every BPA group, there was an increase in mammary cancer incidence compared to controls. This was only significant at the 2.5 dose, the lowest dose, although there's a trend towards significance at the 2,500 microgram dose. Now in the FDA draft report, they have suggested that these results are not significant because when they look at controls from prior studies, the incidence of mammary cancer is actually higher in those prior studies. What is not clear from the draft report, and you have to go look at those prior studies, some of which were conducted 10 years ago, is that the animals in those prior studies were housed in polycarbonate cages and therefore likely have BPA contamination, making them bad controls for this sort of comparison. Low-dose BPA exposure also increased inflammation of the prostate. This again was only seen significantly at the lowest dose of BPA. And this infiltration of immune cells is important because it's a risk factor for cancer. And you're going to hear more from Dr. Gail Prince about cancer in the prostate of these animals. There are also important endpoints that were not reported by FDA in this core study. A prior study that was done just before Clarity as a way to evaluate different outcomes, to help the academics interact with the FDA, and for a 90-day study demonstrated two things. One was that there's a lot of stress involved in gavage, and unfortunately it's hard to evaluate whether the animals in the FDA core study are stressed because there's no report of serum prolactin or corticosterone. These would be responsive to stress, although in webinars, FDA authors of the draft report have discussed these data. So it would be good to see them so we can evaluate how stressed the animals were by daily gavage. There's also no report of serum BPA. And in prior work that was done at the FDA, it was clear that there was a contamination of the animals that were being housed in FDA laboratories. It also is not clear whether the actual source of BPA contamination has been identified, although we've been told academic investigators were notified which of the animals from the academic outcomes may have been contaminated with BPA. There are serious effects of BPA reported in the FDA core study. I showed you the data for two of those. One is mammary cancer, and the other is serious inflammation of the prostate. This contrasts what the FDA has concluded, including in their advertisement for Grand Rounds presentations that are given later this week, which suggested that BPA produced adverse effects at high doses, but not at low doses. And the data simply do not support that conclusion. So why would the FDA ignore the low dose effects of BPA that have been reported in the FDA core study? I think one of the reasons is this expectation that's common in toxicology studies that with more of an exposure, there would be more of an effect. In reality, what has been shown for pharmaceuticals, for vitamins and other essential nutrients, for hormones, and for endocrine disruptors is that there's often a nonlinear relationship between dose and effect. And while some groups have dismissed this as a fringe approach to evaluating chemicals, it's in fact widely accepted by groups including the National Academy of Sciences that non-monotonicity can occur. And there are hundreds of examples in the literature. It's also well documented that there are known mechanisms by which chemicals can act in this non-linear fashion. There's another lesson I'd just like to quickly point out that can come from the Clarity study. So as Dr. Heindel noted, included in Clarity were also animals used as positive controls. These were exposed to ethinyl estradiol, and that's a pharmaceutical estrogen. It's used in many common birth control pills. The Clarity study was including ethinyl estradiol because it was expected that BPA might have effects due to its estrogenicity. A prior study that was conducted in the same laboratory that used the same strain of rats, the same diet, and many of the same housing conditions previously looked at ethinyl estradiol and the effects of ethinyl estradiol on some of the same health outcomes that were evaluated in the Clarity study. Unfortunately, from our analysis of the Clarity study and that prior study that was reported in an NTP monograph, the results of the ethinyl estradiol animals from the Clarity study do not replicate the results that are reported from that prior NTP study. This is true for things as serious as cancer of the mammary gland, which was seen in the Clarity females, but was never reported in a prior study conducted by NTP. What we can conclude from this is one of two things. Either the few differences that there are between the Clarity and the prior study, for example, the use of gavage versus the use of exposure through feed, really are important in dictating the kind of outcomes that we will see after exposures to environmental chemicals. Or, and potentially more concerning, these results comparing these two studies on ethinyl estradiol suggest that guideline studies are actually not reproducible. So there are a few conclusions that we can draw from the Clarity FDA core study. One is that there really were serious effects observed in the FDA core study after exposure to low doses. These have been ignored or dismissed by FDA, likely because they were only seen at the low doses and not at high doses. But this is unscientific based on what we know about how hormones and endocrine disruptors work. We also know that the inclusion of ethinyl estradiol in the Clarity study as a positive control gives us a unique opportunity to evaluate whether or not guideline studies are reproducible. Guideline studies are rarely reproduced because of the large number of animals that they use. The Clarity outcomes looking at ethinyl estradiol did not replicate the effects of ethinyl estradiol that were seen in a prior study conducted in the same lab. I'm now going to turn the discussion over to Gail Prince, who will be reporting information from the academic investigators.
Supposed to go. Good morning, everyone. As has been introduced, I'm one of the Clarity-Bpa independent academic investigators, and I'm going to speak on behalf of the other investigators as well with regards to data that has been published thus far. So as Jerry Heindel already introduced, there were a number of academic investigators who competed in a stiff competition through grant applications to the NIH to obtain the tissues from the FDA and do analysis on end organs that we have expertise in. Our goals were not just to repeat prior studies, because that would not have been funded by the NIH, but to look at novel outcomes and mechanistic insights as well. So what is shown here is a list of the various endpoints that were analyzed in the principal investigators who received those awards. The ones that are in red are ones where data has now been published, and so I can make comments on them in a public setting. My data on the prostate gland has been evaluated and it's soon to be published. So I want to begin by just reiterating what Dr. Vandenberg just said with regards to the FDA core study in cancer, because I look at prostate cancer. And I was very intrigued that in the FDA core study at 1 year, there was a significant increase in breast cancer, and this was most significant at the lowest dose of bisphenol A. Likewise, in the chronic study at 1 year, there was a significant increase in prostate inflammation, which can drive prostate diseases with aging. What I had done in my independent laboratory was a number of things, but I'll just share for the sake of time this one exciting set of data where we looked at the stop dose animals that were developmentally exposed to bisphenol A. And then at the FDA facility, they gave them hormones throughout adulthood that exactly mimic what is taking place in elderly men as they age. Their hormone levels change. And so we wanted to mimic that and look to see if bisphenol A could modify that. The animals were euthanized at 1 year, and we looked at the data. In summary, we were able to repeat our previous findings, and we did find an increase in prostate cancer risk with the greatest effect at the lowest bisphenol A dose. So I'll just share a little bit of that data with you. We looked at 2 endpoints, prostate neoplasia or PIN lesions, which are considered in some instances to be precursor lesions for prostate cancer. In the vehicle animals that had been exposed to these adult hormones, they did have PIN, but it was low grade and not clinically significant. But if they were exposed to estradiol or any dose of BPA, the very low dose, all the way up to high doses, their PIN lesions were now high grade, which are clinically relevant and considered the immediate precursor to developing prostate cancer. And importantly, the greatest severity was seen at the lowest bisphenol A dose. Furthermore, when we looked at another region of the tissue, the dorsolateral prostate ducts, cancers did develop. And they developed across all of the animals, including the controls. But what was striking is that in the lowest dose group, the number of tumors was greatly increased, going up to as high as 8 tumors in the region, whereas the vehicle controls only had 1. So this was a highly significant effect. In a separate set of data, we also found an increase in stem cell numbers only at this low dose. And so that can be tied together to indicate that, in fact, we are affecting prostate cancer as well as the FDA core studies. So the studies combined together give us information that indeed the low doses are increasing cancer risks. Another study that's been published is data on the brain coming out of the Patisol laboratory. They looked at a number of endpoints in the stop dose group. Once again, they looked at the brain structures highlighted up here. These are regions of the brain associated with behavior, gender-specific behavior and anxiety, memory, etc. And these are all established early in development. So they looked at the brains at postnatal day 1 and day 28. And what they were able to find was that they repeated several of the previous findings, not all of them, but several of them. They found an altered size in some of the brain regions involved in these gender-specific behaviors. They found differences between males and females, as one would expect. And they found effects at high doses as well at the very low doses. Very interestingly, at postnatal day 1, when these regions are being established, they found an increase in estrogen receptors, particularly at the very low doses, which will set the stage now for the brain to be more responsive to estrogen throughout life. And they also found changes in other genes that were seen only at the lowest BPA doses. The ovary was examined by the Flaws laboratory, and I just want to emphasize some of her results here. The stop dose study, where they examined the ovaries at different time points, they found a significant decrease in the number of, both the number and the health of egg-bearing follicles at postnatal day 21. That is, as soon as BPA exposure ceased. And this is the peak of follicle and egg number throughout life. Everything declines thereafter. And when they examined at that time point, BPA only at the low doses significantly decreased. not so much in the continuous exposures, although at 1 year there was a decrease in estrogen levels across the animal groups. Heart was examined by the Belcher laboratory. They had limited access for what they could do to the animals. They didn't have an opportunity to give a cardiac stress, but they were able to look at, in the stop dose group, a number of hearts through a number of animals at their hearts throughout development. And they found that there was a decrease in heart weight and ventricle wall thickness at different time points, and they found an increase in the severity of heart muscle damage in the females only at the very lowest dose. They didn't find very many significant effects in the continuous dosing. And so myself, Heather Pattisall, Scott Belcher, and Vandenberg got together and have written a summary of the published effects of BPA that have found significant changes thus far. This is now in press and will appear within the next month. And one of the things that we did was we created what is referred to as a heat map, where we put a box wherever there is a significant effect. Green if it's increased versus controls, red if it's decreased. And we did this across the published studies. And what was striking to us was the large number of reports where significant effects are being observed only at the low dose. So thus far, and certainly all the data is not in, there are 39 endpoints. Half of them are in the low dose group only, 13% in the high dose group, and 36 across both doses. So we applied this same heat map to the FDA core study, and actually we see a similar pattern where there's effects observed significant that the FDA has reported across all different doses. And when we do a similar analysis, we see that a large number of them, 50% in fact, are found only at the low dose group. And this contrasts with what the FDA is claiming, that there's only high dose effects and there are not low dose effects. In fact, if we combine the low dose only with the changes across both doses, it's 70% of the positive endpoints are across those ranges. So overall, at this point in time, we have 75 endpoints that have been published so far with significant effects. Half of them are in the low dose only group. About 80% of them are across both doses. Include the low dose group. So in summary, low dose effects are absolutely taking place in this core and guideline study. And some of these effects lead to adverse outcomes, in particular the cancer outcomes. Thank you very much. And I'll now pass the talk over to Pat Hunt, who's going to pull things together.
Thank you, Gail, and good morning, everyone. I'm going to see if I can distill what we've just heard into major take-home messages, or the major take-home points that we'd like you to take away. I want to start by saying that I also was not a member of the Clarity-Bpa study, but I want to start by recognizing this initiative because it is really a very innovative and badly needed initiative for improving risk assessment. A huge question's loomed over the BPA field for the past 20 years, and it hinges on differences in findings between guideline or core studies and studies from independent scientists. What was interesting about Clarity is the focus was not on trying to determine who's right and who's wrong, but on trying to see if you did both types of studies together, if you did a guideline study, the FDA core study, and independent studies of the same animals, could we in fact determine what was going on and why there were differences? So my first major point is that the power of the Clarity-Bpa study is in the combined data. Now, that analysis hasn't been done. What my colleagues have presented for you is preliminary summaries of some of the data. And from this, it's apparent that there are low dose effects, as they both told you. And I want to come back to this in a few minutes. But first, we have to recognize that Clarity-Bpa is a flawed study. Okay, well, that's pretty much true of every study that gets published, because hindsight's 20/20, and we always realize what we could have done or should have done. But in the case of Clarity-Bpa, some of the flaws actually seriously undermine the conclusions that can be drawn. So I would argue that the strength of Clarity-Bpa is not in what it's going to tell us about the risk that BPA poses for our health, but in the lessons that it provides in guiding similar initiatives in the future. So what are those lessons? The first one is, if the sensitivity of the system is in question, then so are the data. And the sensitivity for Clarity, there are several concerns. One is the animals themselves. There was concern at the outset that perhaps the Sprague-Dawley rat was not the best model for these types of studies because it isn't quite as sensitive as some investigators wanted it to be. But for a variety of other reasons, many of the independent investigators didn't receive the animals they hoped to receive, and their studies were compromised as a result. There were a variety of reasons for this. In some cases, there was a lot of variability among the animals received, for example, in weight. In other cases, there was concern about contamination, and the FDA warned some investigators that some of the animals they received may have been contaminated. Contamination is a problem because it was evident in the pilot study, as we just heard, but the source was not determined, and that means it couldn't be eliminated, but the study went on anyway. And I guess I should back up here and focus on this. And there was minimal monitoring for contamination during the Clarity-Bpa study, but the monitoring that was done did indicate a low level of contamination in some animals. Now, as someone whose research has been repeatedly derailed by environmental contamination, and our latest will be published tomorrow, I know that this type of background contamination can diminish or even eliminate effects. Thus, contamination in Clarity-Bpa may have significantly undermined the sensitivity of Clarity and the effects that were evident in some of the studies conducted. The third concern is stress. As we already heard, the animals were gavaged, daily gavage, and this induces stress. One of the investigators studying the brain could in fact see this because she was given a non-gavage control group. Other investigators did not receive this control, and so they couldn't actually assess the effect of stress on their animals. Okay, despite these concerns, as we just heard, low dose effects are evident both in the FDA core study and the independent study. Obviously, a lot more analysis is needed, and it'll be very interesting to sit down and actually compare across the studies and determine whether these low dose effects are confined to certain groups of animals to see if we can ferret out things like this contamination. But this is actually really interesting because the lowest effects were, most effects were seen in the lowest exposure group, the 2.5 microgram per kilogram per day group. The current tolerable daily intake dose for BPA set by our government is 50 micrograms per kilogram per day. So this is far below that tolerable daily intake dose. And if we were to use the BPA Clarity data, these tolerable daily intake doses are set based on these types of guideline studies. And if we were to use the Clarity data to reset this, and I'm not suggesting that we should, but the newest lowest observed adverse effect level would now have to be 2.5 microgram per kilogram per day because we saw significant effects in Clarity-Bpa at this dose. And this would mean an adjustment of the current tolerable daily intake dose to 0.0025 micrograms per kilogram per day, or 20,000 times lower than the current tolerable daily intake dose. So that gives us a lot to think about. And what I'd like to leave you with is what I think is the lessons from Clarity-Bpa that we can best make use of. First of all, the system needs to be validated to ensure that it has the sensitivity to detect effects if they're there. And that means inclusion of positive controls. This isn't always easy to choose the best positive controls, but it is essential. It means monitoring for contamination. This also isn't easy, but it is essential to make sure that you have the sensitivity that you need for these types of studies. The second lesson I think we need to learn from this is that some of the assumptions that the guideline studies have been based on need to be revisited, because the idea that there's reproducibility among these studies doesn't hold up very well when you look at the Clarity data. It's been assumed that these studies are reproducible. They're not easily conducted. They're huge and they're expensive. But taking controls from one study and using them to assess animals in another study simply isn't a valid approach. And it's clear that the only way to really do these studies well is with contemporaneous controls. And lastly, these low dose effects are a problem because we can't consider these to be spurious effects. The inclusion of high doses to test endocrine disrupting chemicals really doesn't make sense, because like hormones, they can exert powerful effects at very low doses. But the long-held belief in toxicology that the dose makes the poison, that if a little is bad, more is going to be worse, and the expectation that you're going to see a linear dose response is simply not valid. As Laura Vandenberg nicely explained for us, you know, these non-monotonic dose responses are actually expected for these types of chemicals. And lastly, we have to take a hard look at these guideline studies. These are costly, labor-intensive, and they're extremely slow. There's no way that they can keep pace with the emergence of new chemicals. And we need a more facile approach. And we have some wonderful, very tractable experimental models. So continued partnerships like Clarity, where we can partner core study approaches with independent scientists and come up with a better approach to toxicology testing, is essential. The whole really is greater than the sum of the parts. And before I turn this back to the moderator, I just want to remind you that the FDA core study results will be presented in a webinar tomorrow. The URL is on the Green Science website where you registered for this webinar, or you can simply look it up using this title and you'll get a hit right away. But the announcement does state that their data suggests that low-level BPA exposure is safe. I hope that's not the message that we've left you with, because that's not what we think. And what it all boils down to is this non-monotonic dose response. And if you're interested in learning a little bit more about this, I would point you to this wonderful review of why this is the case for these types of chemicals, to bone up a little bit on non-monotonicity. And at this point, I'll turn it back to the moderator.
Thank you, Pat and Gail and Laura. Really great presentations. I'd invite all the panelists to join us with video and audio. And I'm going to start working through questions that have appeared in the Q&A as people have been listening, and invite more people to submit questions to the Q&A. You can see the access to that in an icon at the bottom in the center of the Zoom panel. Several people put comments in chat. I prefer that you use the question and answer panel because I can work with that more readily. So here we have a question. I have to look at what the questions are. From one anonymous attendee, what do we consider, now things are coming in too fast. What do we consider low dose to be relative to what people experience from things like thermal receipts or canned food? Is that relevant to the doses we're talking about here? And I would ask Laura Vandenberg, Dr. Vandenberg, to respond to that question.
You're muted.
You're muted, Laura.
Sorry, it was brilliant what I just said. Based on what we know about human intake from consumer products, the lowest dose that was included in these studies, the 2.5 microgram per kilogram per day dose, is the dose that's relevant to human intake. Children may be exposed actually to even a little bit more than 2.5 micrograms per kilogram per day. Estimates have intake at even up to 10 micrograms per kilogram per day.
We have another question. Will the webinar recording be available afterwards? Yes, it will. We'll have to do a little bit of work on the video to clean up the beginning and the end, but it will be available at the site where you registered as soon as we can do it. And I would expect within about an hour after we close the webinar. A question as to why did the FDA use historical controls instead of the study control animals? And I would ask any of the panelists to respond to that.
So I can take that. It's actually somewhat standard practice in regulatory toxicity testing to have a series of controls from prior experiments. This would be important, for example, if a study of a novel chemical found a very unique outcome, something that's usually not seen in animals, and you would be able to go back to those prior controls to show just how rare that tumor is. So in your core study, you might find a rare kidney tumor in 1 out of the 50 animals, but you would want to go back to the historical literature to show that the usual rate of that rare cancer is 0.001%. That would raise your concern about seeing that rare tumor after exposure to your new chemical. In the case here, it seems that the use of historical controls was not done according to the National Toxicology Program's actual guidance. They used historical control animals from studies that were quite a bit older, which is not according to the guidance. And they used them as a way to try to suggest that mammary tumors are actually quite common in this rat strain. But when we look carefully at the studies that were used to generate those historical controls, those studies used animals that were housed in a BPA-containing environment, polycarbonate plastic cages, for example. So they used them as a way to try to diminish or dismiss the importance of the mammary cancer that was seen, but I believe that that was not actually scientifically valid.
Thank you.
If I could add to this, one of the mistakes we frequently make is to assume that a strain is a strain is a strain. And of course, genetic drift can occur over time. And so this is one of the concerns and one of the reasons for contemporaneous controls is we really can't control for natural genetic changes that occur over generations.
Anyone else wish to contribute to that? So we have a comment from another Clarity participant, Dr. Thomas Zeller. He says, he reports to the group that a point of interest to listeners is that the European Commission has recently invested over 50 million euros in a program called Horizon 2020, and several of the funded projects are using the Clarity design. Thus, he says, internationally, the Clarity design will continue to be important. Thank you, Tom. So here's a question for Dr. Heindel. You pointed out that the guideline studies are government approved, but your presentation implied that independent scientific studies are not. Given that NIH, NSF, and EPA, if not all federal agencies, fund projects that have been reviewed by several groups of government and independent scientists, did you intend to imply that the independent studies have not been government approved?
So that's an interesting question. The difference here is that the guideline studies have good laboratory practices, and their design is proved by regulatory agencies for use by industry and other regulatory agencies. They approved the design and the endpoints. So that's different than the independent scientist studies who design and do the studies based on hypotheses they develop in their laboratory. And I think that's the main difference. Does anybody else want to comment on that?
As one of the Clarity investigators, I just want to reiterate that, in fact, the studies that we proposed and that we carried out were in fact approved at a peer review level and by the NIH when they awarded our grants. And so the peer review, they didn't just hand out these grants to whoever asked for them. It was a stiff competition, and it was analyzed by scientists and the government officials working at the NIH to be a valid, scientifically valid approaches to be utilized.
Great, thank you. Here's a question. What evidence is there that gavage administration is stressful? And why was the possibility of high stress not determined in Clarity?
I think there's actually an extensive literature that shows that gavage alters stress hormones. In fact, I'd be happy to put a link to anyone who would like to see that, a paper that reviews that. But specific to Clarity, I mentioned this sort of pre-Clarity study that was done as a feasibility study. And part of that pre-Clarity study included non-gavaged control animals, so unhandled animals, and gavaged control animals. And
Laura,
they were different. Yes.
I think some people would like to know what gavage is.
Oh, what's gavage? Yikes. Gavage is when you restrain the animal with your hand or in an apparatus and stick a tube down its throat so that you can put the chemical in a solution into the stomach of the animal. So it's a way to absolutely ensure that the animals have been exposed because you know you put it into their stomach, but it's not how humans are exposed unless you're unless you have a feeding tube.
So it is a stressful procedure.
Yes, but as I noted in my presentation, the data that would actually allow us to know how stressful is not available in the core study report.
Thank you. Anyone wish to add to that? So here, another practical question. How does BPA leak? Is it directly by handling products or by heat?
So I'll take that one since I seem to have too much experience with this. Any plastic that can be damaged will start to leach the chemicals of which it's comprised. And we've had repeated bouts with this problem in our animal facility, try as hard as we may to be careful about everything. But basically any plastic that's showing signs of wear and tear can leach. Similarly, that plastic resin that coats the inside of food and beverage containers is a potential source of leaching chemicals. And as we've learned, particularly oily chemicals like spaghetti sauce will tend to pull the chemicals out of that resin, thereby contaminating the foods. So that's at least one way. Similarly, those pressure printed receipts we all handle on a daily basis have a matrix that contains bisphenol A or replacement bisphenols, and simply handling them is a way to get exposure. Extra bonus points if you're eating greasy French fries or have just coated your hands with hand lotion, because that enables more of the chemical to get in.
I just might add there that there are thousands of products that contain BPA, and it's not just in plastic bottles and cans lining. BPA is one of the highest volume chemicals produced in the world, and the industry has found thousands of uses for it. So we are surrounded by it. The CDC has confirmed that the majority of humans, well above 90%, have BPA in them at any point in time, even though we know that BPA leaves the body very rapidly. So how it gets there is not completely clear all the time, but it is there, and that has been confirmed by government testing, I might add.
So another question, the FDA and academics were supposed to be evaluating the data together and co-publishing these results. Given that, what are the expectations now for the integrated results?
Well, as one of the investigators, there are a number of approaches that are being utilized. I know that the National Toxicology Program, the NTP, is waiting to get everyone's data, and as well as the core study report from the FDA, and then we'll go through with experts and scientific rigor and make a determination. So this is yet coming out in the future, and we have not yet reached that stage. There are other joint publications and analyses coming out. Jerry Heindel is helping coordinate a group effort of the individual scientists to give summaries of the results in a publication so that everything can be published together. One of the issues that we recognize as the independent scientists is that we're all publishing our data as we're finished with it in a variety of journals. And so there's not a singular voice coming out comparable to the singular report at the FDA. And so recognizing that we're working on making a combined report. But certainly the head of the NTP, which is a branch of the NIH, is going to be evaluating things as a whole.
Anyone else wish to comment? Okay. Different type of questions. What actions, if any, do you recommend parents take while we wait for this scientific debate to settle?
It's not an easy question to answer. I mean, I think that as a scientist, I sort of embrace the idea that science itself is never done. And we shouldn't wait for all of the answers to make decisions that are better for ourselves. It's part of being precautionary, but it's also just part of using the science that's available. We know from more than 100 epidemiology studies that people who have more BPA in their bodies are more likely to have adverse health outcomes. That doesn't mean that we can show that it caused those health outcomes, but I think we have enough science to act. To me, what that means is taking the simple changes that you can make in your life, reducing your use of canned food, don't handle thermal receipts, don't microwave in plastics, reduce your use of plastics entirely. But we also have to recognize that not everyone can afford to make those changes, that some people, their major source of vegetables is canned food, and we don't want to discourage people from eating vegetables if that's their source. We also have to acknowledge that going towards BPA-free products has led towards the increase in exposure to BPA-like chemicals, bisphenol S, bisphenol AF, bisphenol F. And other phenolic chemicals are being used to replace BPA in a lot of consumer products. For me, I think what that means is that we need change at a much higher level, and that we need data like what are being produced in Clarity to actually lead to change so that we don't have to be chemists when we go to the grocery store to make purchasing decisions.
I'd just add to that I always encourage consumers to start thinking about plastics differently. I'm on a college campus and I see college kids walking around with their sport bottles, and some of those sport bottles look like they've been walking around with the same bottle for 10 years. There's plenty of signs of wear and tear. But I encourage people to think about these not as durable products, but as disposable products, because if they're showing signs of wear and tear, they are leaching chemicals. And like Laura, I always advise that heat is an invitation for chemicals to leach from these products. So microwaves, dishwashers, are an invitation to pull these chemicals out.
Okay. A anonymous attendee is asking, do you think these findings are applicable to the replacements used in BPA-free products?
The data that's come out on products that have a similar chemical structure, as was just mentioned, BPS, BPAF, others in the bisphenol family, have found pretty much identical, in some cases actually worse results. So the advertisement of BPA-free, one needs to be cautious about, because they could have easily been replaced with other chemicals that have the same characteristics.
And I would add to that that we've, looking at germ cells, the cells that are going to make eggs and sperm, we've seen the same effects as we see with BPA with several of these replacement bisphenols. And our latest inadvertent exposure was in fact to one of these replacement chemicals, BPS. So yeah, I fear that these are not safe replacements.
One of the people in the audience is not convinced by the discussion about potential contamination and would like more discussion of why you think there was contamination in the control animals.
So this is interesting because contamination, background contamination is problematic. And so you can have contamination enter your system during the collection of tissues, during the processing of tissues. In this case, we know that the animals were exposed for one very simple reason. If animals ingest BPA, they metabolize it. And if you see metabolites in the blood of animals that's collected, you know the animals themselves were exposed, not just the tubes you collected the blood in. And that was the case for the pre-study for Clarity-Bpa.
So this information is coming from the pre-study that was published. In the Clarity-Bpa itself, they did not collect the serum after the BPA exposures and measure it using techniques that could look at the BPA metabolite. And so we do not have actual information from the Clarity-Bpa study because it was not collected in order for us to have that. That said, the previous study had indication that some of the animals may have been exposed and to a background level because they had this metabolite in control animals that should not have had that metabolite. The degree to which that occurred in Clarity is really uncertain. One of the explanations that the FDA gave was that, but it was not proven, was that the co-housing with animals that had very high dosing may have crossed over in the air or in some other device and gotten into the control animals. And we do know that a good deal of some of the Clarity studies themselves, the animals were also co-housed with those very high dose exposure groups. And if that indeed was the source of contamination, which we don't know, then that possibility would remain. So it is a question mark. We don't know it for certain because it wasn't measured.
Thank you very much. And I'm afraid we've run out of time. I'd like to thank the panelists for giving a very coherent overview of Clarity-Bpa. And also, I'm very grateful that we have many participants from around the world and you've submitted excellent questions. Before we close, I'd like to alert you to a new scientific publication that will appear on the website of the Journal of the Endocrine Society 2 hours from now. The Endocrine Society is the medical and scientific professional association of the world's endocrinologists. These are the doctors and the research scientists you go to when you have breast cancer, prostate cancer, type 2 diabetes, or other disorders that involve mistakes that hormones are making in regulating your bodily function. This study is from the Von Saal Laboratory at the University of Missouri and is the first randomized experiment with people that measures the physiological effects of BPA delivered orally at the reference dose. It is a small exploratory study that must be replicated. The results though, would be impossible if the FDA and EPA's current safe dose is correct. So thank you all. We will have this tape online as soon as possible.